morning everyone. In this video, I'll be presenting my project on protein secondary structure prediction. The goal of my work is to accurately predict the secondary structure of proteins by combining different approaches. The first approach is Chow-Fasman, which is actually a rule-based approach. The second approach is GOR, which is a probability-based approach, and the last approach which I have successfully completed is Blast-in Predator, which is a homology-based approach. At this stage, all of these three approaches are completed and working successfully. Here is a quick overview of what I'll be covering. I'll start with an abstract, then I go to the introduction part, followed by the objective of my tool. Then I'll explain the background and methods I have used. After that, I'll show the current status of the project, give a summary, and end with the references I have used. Introduction. Proteins have structure like alpha helices, beta sheets, and coils, which are known as secondary structures. They are critical for protein function. By predicting them directly from amino acids, we can make big strides in drug discovery, protein design, and bioinformatics. In this project, I have combined biological models with modern data-driven techniques to improve prediction accuracy. My objective is to build a tool that can predict protein secondary structure using Chow-Fasman for rule-based prediction, GOR, to correct these predictions using surrounding sequence context, and Blast-in Predator to validate the predictions using similar known sequences. I also plan to improve datasheet prediction using a method called as beta program, and also implement 2D and 3D visualization as well using Matplotlib and other tools which are available. There are two main approaches to predicting secondary structure. One is experimental, in which we use lab techniques like X-ray crystallography, NMR, and cryo-electron microscopy. The other is computational. The one which I have been using in computational is statistical models and probability models. The other techniques which we can use are machine learning and deep learning. The main reason why I shifted to statistical and probability models are machine learning and deep learning are a bit tough to implement and needs a very high knowledge to complete it. Let me briefly explain the methods I have used. Chow-Fasman predicts the secondary structure using amino acid propensities, whereas GOR adds context by looking at nearby residues and using information theory. Blast-in Predator compare the input sequences to known structures to find matches. Later I'll include the beta program and other things, which uses basically hydrogen bond rules and other geometries. The methods which I have mainly used are, at first I use Chow-Fasman, which is one of the earliest statistical models. It uses known propensities of amino acids to form helices or sheets, identifies where other structures might begin. From there, it extends prediction based on scores of neighboring residues. It's a solid starting point, but it doesn't account for long-range interactions. So the accuracy is pretty much convincing, which is around 50%. The second method which I have used is actually GOR method, which is based on Bayesian probability and information theory. It considers a window of 17 amino acids and calculates the probability of the center residue forming a helix sheet or a coil. I implemented two versions of the GOR. The first version is basically the existing GOR matrix which I have taken from the GOR1 research paper, by which the accuracy was pretty low, which was around 42%. So I shifted to a custom made GOR matrix using dataset from Hugging Face, which reflects more of recent protein data, where I'll be tracking the sequence and also its secondary structure and also creating the matrix, which had gave a pretty good accuracy around 60%. Using both gives my model flexibility and relevance to model sequences. The third approach I used is homology based. I took an unknown protein sequence and used BLAST to find similar sequences from the dataset, which I have mainly taken from the Kaggle website. Then I used Predator to extract the secondary structure from those similar sequences. If a good match is found, I use the structure. If not, I will fall back to Chow-Fasman prediction. This method adds an evolutionary perspective which really helps improve accuracy, especially in conserved regions. I'm currently working on two more components, which are mainly beta program and also 3D rendering using tools like Pymol and Camera. For visualization, I plan to include 2D plot which was almost completed using Matplotlib and the 3D rendering using Pymol and Camera. Beta program will focus on specially improving beta sheet prediction using hydrogen bond patterns and geometry. So far, I have completed the Chau-Pascal model, which is rule-based and achieved 50% accuracy. GOR model implemented both original matrix and also new custom matrix using modern datasets. Together, I got accuracy around 60%, which was pretty good compared to Chau-Pascal accuracy. Also, I have implemented the blast and predator modules. It performs homology searches, retrieves similar sequences, and uses them to make structural predictions. If no match is found, the system automatically switches to Chau-Pascal. The accuracy which I have got for blast and predator is around 65%, which was better than Chau-Pascal and GOR. I'll be further trying to improve the accuracy of Chau-Pascal and GOR.
So now I begin with Chau-Fasman method by defining a dictionary of propensities. This maps each amino acids to its known alpha helix, beta sheet, forming potential based on empirical studies. The propensities help identify which amino acid is structurally biased towards forming helices or sheets. In the function, I initialize all residues as coils by default. Then I iterate over amino acid sequence using a sliding window of 6 residues for alpha helix and 5 residues for beta sheet prediction. For each window, if a sufficient number of residues exceed the predefined alpha propensity threshold, I mark those regions as helix and continue extending it until the local average drops below threshold, which we can actually see here. The evaluation metrics which I have used are accuracy, precision, recall, and F1 score. I took the sequence and also its true structure from one of the tools of Chow-Fasman available in the internet, and I predicted the accuracy, which was at most 50, and also the precision was 44%. Similarly, I perform a separate phosphate beta sheet detection using beta propensities and 5 residue window. If the region qualifies as beta sheet, I labeled it as E, but only if that region hasn't already been marked as helix. That hasn't been marked as helix. This avoids the structure overlap. The final result is a string of secondary structure labels: H for helix, E for beta sheet, and C for coil, returned for full sequence. The second method is actually GOR, where at first I took the dataset from the Hugging Face, which I imported and loaded, and the dataset is loaded. The implementation starts with the statistical approach where I build my own probability matrix from real protein dataset. I load the sequence and their known secondary structure using the Hugging Face protein a secondary structure prediction on dataset, which provides amino acid sequence and DSRP labels for HEC. Using a sliding window of 17, Using a sliding window of 17, I collect the frequency of each amino acid at every position relative to the central residue grouped by the actual structure label. So for each structure type, helix, sheet, coil, I build a frequency distribution for amino acids across the window positions. After collecting counts for all training samples, I normalize the frequency into probabilities. This forms GOR probability table. For prediction, I again slide in the 17 length window across the new input sequence and compute the log probabilities of the central residue being in each structural class. This is done by summing the log of probabilities of the observed amino acids position by position across the three structure types. The structure with the highest log probability is assigned to that position. The evaluations which I have used are actually Q3 accuracy, per class accuracy for H, E, and C, and also the Matthew correlation coefficient MCC, and also confusion matrix for visualizing by using Seaborn. The accuracy which I have got for the GOR is almost 60% with Matthew correlation coefficient as 38%. Per class accuracy is for H I got 65%, per E I got 60%, and for C I got pretty low 53%. Along with the data-driven GOR model, I implemented second version using original matrix from the published literature. These predefined score tables that assign weights to each amino acid at each of the 17 positions in a window for each of the three secondary structure types. Structure here is slightly different. Each amino acid has a fixed 3 into 17 matrix, one row for helix, one for sheet, and one for coil. During the prediction, I paired the input sequence with the dummy residue to handle window edges. Then slide the 17 length window across for every window. I calculated a cumulative score for each structure, the type by summing the scores defined by the matrix for each amino acid at positions. The structure type with the highest score total for that window becomes the predicted label for that residue. This approach mimics the original GOR1 algorithm and requires no training phase. Since it relies on predefined matrices, it is deterministic and useful as consistent baseline. Lastly, I built a homology-based prediction method that mimics how Blast and Predator works. I load a local data set from CSV file that contains protein sequences and their corresponding secondary structure annotations. Here we can see the PDB ID, sequence, SST8, SS3, SST3, and length, and all other necessary data. SST8 is actually a secondary structure sequence with 8 components like H, E, C, S, T, etc. SST3 is mainly the thing which we are using, which contains H, E, and then C. For a given query sequence, I compute similarity scores with all known sequences in the dataset using sequence matcher, which gives a ratio representing how closely two sequences align. Then I select the top three sequences that have similarity score above the threshold. Once the top matches are identified, I align their secondary structure annotations using back to the query using block matching for sequence alignment. For each position in the query, I perform a voting mechanism. If 2 out of 3 matches suggest helix at the position, one suggests sheet, and the helix gets more weight. Importantly, the votes are weighted by sequence similarity scores, meaning more similar matches have more influence on final prediction. If no similar sequences are found, I use a fallback mechanism that triggers the Chow-Fasman predictor to still produce an output. This ensures the system is robust and does not fail when homology information is sparse. After prediction, I compare the result to the reference annotation and compute the match percentage to evaluate how well it performed. The accuracy which I have got for the Blast and Predator is pretty good when compared to the other two, which is 63%. At last, each of the three models I implemented contributes uniquely to overall pipeline. Joe Fassman provides a fast, rule-based estimate. GOR introduces statistical learning from biological data, and original GOR matrix preserves historical accuracy. Finally, Blast and Predator brings evolutionary information and alignment-based validation to the table. Together, these make the system more accurate, flexible, and capable of handling a wide range of protein sequences. That's it.
To summarize what I have built, a combined system using Chopasman, GOR and BLAST for predicting secondary structure. Chopasman gives me a fast and interpretable rule-based baseline but has a little bit less accuracy. GOR adds statistical contents. Blast and Predator introduces evolutionary insight. These three main methods complement each other very well. I'll be adding beta program and visualization tool next to make the system even more complete. Here are the references I have used. The original Chopasman and GOR papers, Blast and Predator documentation, Hugging Face for the dataset used in custom matrix generation, and Kagan for sequence similarity data. These sources helped me build and validate my model. Thank you.